Welcome to the roundtable of this afternoon, which tackles a very important matter, which is concerned by the reason for which we all work. I think, in addition to the basic science that we do, is that how can we best transfer from bench to bed? Best is an important matter, but even if it would be just transfer, it would already be quite an important matter. And for this very important matter, three experts in the field have gathered, and I will try to moderate this session. We all think that the session is of enough importance for all people here and should really be a roundtable, that we should try. I know it's not easy in an amphitheater like this one, but we should try to organize a roundtable not only between ourselves, but also with you and the audience, and that we all feel free. We are at the end of the day. We all feel free to interrupt, disagree, agree, give experience that we have and you have, and try to make it a lively discussion. Indeed, it is organized in a way that there will be no speeches, but there will be questions, which, and I would like to pay tribute to the members who are here, who have prepared very carefully that, and these questions will be introduced and discussed by various examples, and of course, we will put forward the examples that we know best, but I am convinced that you have things that may be directly useful to the discussion and useful for everybody. So my role will be to try to look in the audience each time to see if some of you want to say something, and I know enough people here to know that there are people from academics, from industry, from valorization, from different parts that should make the discussion lively. So maybe, of course, it's not necessary to introduce the people who are around me, but still, before we start, it's always useful to know from where people are speaking, and I could start by a very short introduction of myself first, since I am the oldest around this table. My name is Wolfgang Friedman. I am an immunologist mostly involved in tumor immunology and in the analysis of the ways the local immune microenvironment controls cancer development, and how it could provide markers for prognosis and maybe for indicating response to therapies at various levels of cancer, and we could discuss it later. Maybe John.
Thank you. Yeah. Okay, great. So my name is John Connolly. My research interests are in dendritic cells and rational vaccine design. I've also got a keen interest specifically in human immunology and understanding the changes in academic structure that are required to really facilitate true human immunology research. I'm in Singapore, Immunology Network, sorry.
Okay, my name is Marc Bonneville, so I'm a cellular immunologist. Actually, I'm not really a specialist of downstream implementation of immunotherapy trial, but actually several teams in our research center in Nantes have been working in this area for many years. And And so based actually on this overall experience of the non-team, I will address some specific point regarding implementation of these trials.
And my name is Matthew Albert, and I'm at Institut Pasteur, and it's a real honor to be part of this panel. I should say that I've been dealing with this question for the past 18 years throughout all of my training as a MD and PhD, and it's a very schizophrenic existence. And it's interesting to figure out how to translate from one side of the brain to the other side of the brain. So we have a very special challenge in front of us. I wanted to start with one slide before we jump into the questions that the panel has identified. And this is a slide that I've been showing over the past 5 years as myself and colleagues at Institut Pasteur, in particular Lars Roge, have developed a Center for Human Immunology to try and address exactly some of the hurdles that we're going to be talking about for the next hour and a half. So we are very aware of the gap that exists between mouse and man, and it has been a point of special interest for immunologists, because immunologists seem to love their model systems, and in particular, and this is really a tribute to Philippe, who is responsible for training many people in this room. In my case, I did not have the pleasure and honor of training with him, but he is responsible for my being in France, and I do thank him every day for that. He's also responsible for helping to inspire a lot of our models on infectious ovalbuminemia and the use of ova in many experimental systems. Unfortunately, we have been misled in that regard, in that very few examples exist where what we have learned from our model systems have really translated into important information regarding chronic infection, tumor immunity, autoimmune responses, transplantation, and allergy. And for those of you who do not know, Philippe is responsible for the initial cloning of the chicken ovalbumin gene. And we do celebrate his contribution to immunologic models and the special challenge he's created in translating this information into human. So, as I mentioned, the committee, the roundtable group has identified 4 or 5 questions, and if it's okay, I'll just lead I'll lead with the first one. And we have identified operational infrastructure issues as one of the hurdles involved in facilitating the translation between bed and bench or mouse and human.
Okay, so I will introduce this part, and I would like to mention actually several key issues that should be taken into account when implementing an immunotherapy trial regarding particular technical, logistical, and regulatory constraints. So the first point is that you need to get the right infrastructure. So the right infrastructure is what? Is a cell therapy facility, it's a clinical investigation center, and more importantly, it's a whole staff actually that is well trained in quality assurance and regulatory filing. And I should insist that this is the one key bottleneck here when implementing immunotherapy trial, is to find actually a dedicated staff that can fill in all these regulatory forms. The second point is that when you are implementing a cell therapy trial, you have to design a fully clinical grade process. So this must include exclusively GMP grade reagents. And here, sometimes it's a real challenge, actually, to find the right reagent. It's expensive. They are not always available. And so this raises an issue regarding, actually, the facilities that could provide this kind of reagents. And more importantly, such reagents might not be suitable for optimal expansion or differentiation of immune cells if you are dealing, for instance, with cellular immunotherapy. So the point here is that sometimes it can take several years before designing the right process that should be as reproducible, simple, cheap, and efficient as possible. So just to illustrate the kind of trivial problem we have to solve when designing this kind of process, For instance, in Nantes, there are several teams that have been working actually on implementation of dendritic cell-based or T-cell-based trials. And the problem here was to find, for instance, a GMP-grade reagents, either fully defined media or a clinical-grade sera that could allow optimal expansion of T cells or differentiation of dendritic cells. And actually, after several years of investigation, we came to the conclusion that probably the best solution was actually to use clinical-grade human serum batches that were specifically screened for these purposes. So it's a kind of very trivial problem, but the problem is that you have always to interact permanently with regulatory agencies to see whether it might be possible actually to generate this kind of human sera, whether there is a probability that, for instance, this clinical grade product will still be agreed in 5 to 10 years. So it's sometimes very tricky, and generally it requires a concerted action either at the national or international level between teams working specifically on these issues.
Marc, can I interrupt you? So do you think we're going too far in terms of regulatory issues related to GMP? I remember the first dendritic cell adoptive therapy protocols where they used fetal calf serum. Yeah. They showed incredible benefit. And in fact, it took many years to discover that it was the LPS within the FCS that was critical for DC maturation and some of the potency of that therapy. Are we not? Are we going too far as from a regulatory standpoint? Should we be fighting regulators to minimize some of the stringency on GMP and GLP?
I think that these are two different worlds, and the best way actually is to try to find a common language. So the best way actually is to put more scientists actually in a regulatory review panel, so that actually you can expose actually your options, and actually, well, the people even in regulatory agencies are quite pragmatic. And if they consider that the risk-benefit ratio is favorable, they might be willing actually to accept sometimes some optimal golden medium situation. So I think that it's just a matter of language and discussion, but it's a lengthy process. So you spend a lot of time actually going back and forth with it.
To stay on this level, I know we will have a discussion later on the place of industry in this, but I would like to have a comment maybe by Jean-Pierre Abastado or his neighbor, Margarita Salcedo, who had to tackle exactly the same question, but from the other side, in a company who made dendritic cells, cell therapies. And I remember that it was the pivotal question. The idea was even, we will succeed because we will be the best in process, because everybody will have to pass through this process, and you have had a lot of discussion with the regulatory agencies. Do you have a comment at this level?
Okay, maybe I can shout. The problem is not only the agreement from the regulatory agency, but eventually if you develop a drug, at some stage you would like to make it on a large scale. Yep. On manufacturing is one of the main cause of failure of drug. So although I think having a human serum which is screened for its property of inducing maturation at some stage in the development of your drug, you will need to have a maturation agent which is easy to manufacture. If you have to test, to treat maybe 30 patients, maybe this is okay. But if you have to treat 3,000 or 30,000 patients, you need to have a drug which is easy to manufacture. Serum is a very difficult product.
Sure.
On the choice that we did, in fact, addressing the exact same question at IDM, was to use a drug, ribomunil, which was already approved and manufactured by Pierre Fabre, which we could use as they would do the manufacturing, they would do the quality control, and we would have a drug for which all the manufacturing issue had been solved by somebody else. To get the approval from the regulatory agency is one step, But being able to manufacture the drug is something really very challenging, especially when you move from phase 2 to phase 3.
But yeah, with respect actually, for instance, to the human serum, when I'm talking about, for instance, the possibility to make large batches, it's not academic or even particular private company that would do this. For instance, it's the blood center that would have actually all the GLP facilities to do this kind of large batches. But the point is that this kind of batches must be screened for this purpose. And this is the key point. And I think that this is really, for instance, a limiting factor, for instance, for cellular immunotherapy, because what we realized is that in several trials where we perform some adoptive transfer of T cells, the number of doubling in vitro was key actually to the later on to the in vivo survival and the therapeutic efficacy of the process. So if you have suboptimal, although GMP grade condition for expansion, for instance, of T cell clone, actually the efficacy of your product at the end actually might not be so good. So I just wanted to mention another important point is that also what is very important is the phase 0 trial. Feasibility trial, because generally what we do is we assess actually all these processes in healthy donors. And the problem is that, particularly in the field of immunotherapy, when you actually translate this to the patient situation, the problem is that frequently your patients are immunodepressed. And because of, for instance, several immune escape mechanisms, even, for instance, the purification, trivial aspect, purification of T cells, for instance, is very different. You have much more contaminant that can be immunosuppressive and all this stuff. So this is one point, this is one limitation, but also what is very important is here, this introduced significant bias in terms of patient inclusion. And the problem is that once you start screening, for instance, for patients that show some immunocompetence, you have to be aware that the immune status of the patient is a positive prognostic factor in various oncological situations. So then this really hamper actually the assessment of the therapeutic efficacy of your protocol. So it's another important point. The last point is, but you introduced it, Jean-Pierre, is the logistical issues. Should we perform a mono versus multicentric trial in order to increase the recruitment? Should we use frozen or fresh samples? Should we inject frozen or fresh cells? Should we design closed or open system? And all these key issues actually have a dramatic impact on the feasibility and length duration of your trial. Next.
Yes?
No, I think the problems that we are encountering to industrialize this kind of products have been already mentioned, and I think one of the key also tools for success is that you have quality controls of your product that are reliable and are consistent, because then you will control in that way also your ancillary products. If you don't have very good quality controls that are telling what the product is doing, especially, for example, cells, then you can inject something that is not reliable. And so I think all of this together has to be taken into account when you want to translate something, because these quality controls are based on mechanisms of action, but they have to be simple as well, because if you have large-scale trials, you have to do quality control of your product that are simple, but that means something. So I think this is a central issue as well.
Getting back to this concept of educating the regulators, I don't know what the regulatory authorities look like that you guys have used on your cell-based therapies. Jean-Pierre, I do with your trials, but they really don't, they certainly aren't educated in concepts of QC around, dendritic cell responsiveness, and that there may be a need to even set up, if possible, a secondary regulatory authority or a parallel regulatory authority that at least is educated enough to evaluate some of the key parameters for cell-based therapies, particularly in immunology. One thing you got probably from that the last presentation is that it's extremely difficult, and in somewhere within this product development pipeline, which we just saw there, there's a real opportunity to do some real science and do real research. And so I look at this field of translational immunology from two standpoints, really. And the first is that the first is really from a medical standpoint, is how can we utilize what we know and the knowledge about immunology to cure human disease? The other is how can we use the existing medical infrastructure and the ongoing clinical trials that are going on, which have regulatory endpoints, to actually do real experimental medicine in humans, to do experiments in people to understand the human immune system. And to really do that, and for an immunologist to then step out of the mouse room and into the clinic, you absolutely need real support. And the way we've approached that is to take it from an institutional standpoint and to look and identify the gaps that exist within the entire translational research infrastructure. So from the scientist, from the support for the scientist, from the academic support in the institution, its history, its habits, all the way to the clinic and what the physicians do. And so what we see here is that this is a clash of cultures, right, with completely different mandates, and that this largely inhibits most of the translational research that's ongoing. Thank you. So what we've done is we really looked at this not from the standpoint of someone that wants to, so I want to get my dendritic cell therapy or my new rationally designed vaccine into clinical trials, what do I need to do? But rather more systematically and ask, what do we all need to do to improve this pipeline of translational research from, so that someone who's worked their entire life, trained in mouse research, can then move immediately into people. And so we identified three, I think, key components that are missing within the current translational research and medical and academic environment. The first, and one of the most daunting problems, is that you have within, at least in the US and certainly in Singapore as well, medical community whose central mandate is absolutely positively not research, and nor really should it be. So it's really, they need to have infrastructure and training programs that can support them. And this is one of the major pieces that's missing, and this is true low-hanging fruit within the healthcare system, is translational relational databases, relational clinical databases. So this is an ability for the physicians in the clinical groups to actually organize their data on the patients that they treat in a way that allows us as scientists to actually ask reasonable questions. You may all know, and many of you are, I'm sure, practicing physicians, that just asking someone to go and look up, the medical results on some of the patients takes absolutely forever. And so if there's a way to actually organize this data, this is actually quite cheap and something we can implement. The other is actually supporting research nurse staff, so dedicated clinical personnel. This is a major issue that can be addressed with relatively low contribution. And then again, there's also a preclinical support network. So this is people that can actually work on things like IRBs and FDIIDs. When you approach a clinical team, I'm not sure who you think is going to do this, or who I think is actually going to do this. We need to create dedicated staff that can work on this, and so this absolutely has to be in place. The other missing component that is true for both the research side as well as the clinical side is rational biobanking capabilities. So true biobanking science is essentially missing from both the academic environment and in the medical teams. And so aside from a few excellent biobanks in Moffitt and in the UK, what you have is basically surgical teams that have -80 freezers that they've collected over years that they call a biobank. And what you really need is a true biobanking system, which is a collection of the clinical data, the biobank itself, and the IT infrastructure to pull this together. And then finally, something that I think is important, I think is really emerging as a central, probably out of the regulatory field, is this concept that you need central facilities to assess immune status, and that centralized core facilities are really the place that can do this in a reproducible, high-quality way. And so this is really where this immunomonitoring concept comes in. And so if you put these three components in place, what you have then is a pipeline that the research scientists can engage the physicians in a meaningful way to move a project forward very quickly.
Any comment on this building, which seems to be quite a perfect, oops, it has disappeared, but still quite perfect building with... I have one question about that again. It seems that being perfectly organized institutionally. But we know that many of the novel drugs or approaches or modulators will come from something outside of this institution. So how do you look at the interfaces between this Greek temple and the fact that it is open it should be open to different winds and should accommodate and integrate that?
Sure. So I think that what we're really talking about is essentially filling gaps that are missing, both with new drugs that come on board or new concepts that are coming out of the immunology field. I think that what we can do is address that by building innovation into the platform. So with regard to, say, the immunomonitoring platform, technology development and new technology and new assessment has to be an important part of all of this. So, things that are critically important in GLP labs. This actually, I mean, we talk a lot about GLP and immunomonitoring as being critically important, but actually GLP certification gets in the way in many ways of discovery, right? So we can monitor, say, a phase 1 clinical trial using the infrastructure components that you see here, but what you miss by doing completely GLP monitoring is the ability to look at that next new marker on an important disease subset. And so this is really this innovation that you're talking about. So, and I think that this highlights something that I think is critically important. Everyone, I think, in this audience can look at that diagram and say, it's great if you had $50 million and, an enormous army of people to actually approach it. But this is not really what you need. What you want to do is look at the ongoing clinical trials that are out there right now in the community and ask, how can I build a research arm onto that clinical trial, and what do I need? And this is not a huge amount of money to ask simple questions, to get in on the ground floor, to try to work the key time points in which you can sample these patients so that they still meet their regulatory endpoints, but you can now sample them and look at them scientifically. And to do that, you really do need this infrastructure, and this infrastructure has to be flexible. You need to look at that next new marker on those patients.
Which is not easy, because the problem is that when you are a very well-organized biobanking structure, actually it becomes extremely difficult and expensive actually to actually get access to the sample, because generally the size of the sample is quite limited and there are many people who are asking for, so it remains actually an unsolved issue.
So this is a key area of technology development. We talk a lot about, now that we've reached this immunomonitoring part, we talk a lot about technology development being an important part of this, but almost all of the technology development is cost reduction and sample size reduction. You spend all of your time saying, how can we do this cheaper with less and get just as much information?
So it's quite funny to be on the panel with Marc and John. It's okay. John in particular, because we have a very common scientific starting point, which is dendritic cell biology, and have sort of evolved sort of in parallel towards translational research. We have a very different and I think unique situation at Institut Pasteur, which is where I'm based. I know that there are many people in the audience. And when it's been, I think, just over 10 years that we closed our hospital, and it's been a question of sort of how to maintain a strong medicalization and translational approach, something that was obviously a founding principle of the institution. And as I mentioned in the introduction, a number of us have gotten together over the past years to create a Center for Human Immunology that started 5 years ago with 6 projects. Half of those had real clinical partners and were bringing human subject samples into the institution. We now have over 25 projects, 11 of which are dealing with human subject samples. And one of the things that has been very important for us, and I think an area where we've really offered added value to the community, is by providing very strong technical support. That is both interesting to the scientists who are doing sort of basic and preclinical research, but also of value to people doing translational research and working with human subject samples. And I just want to highlight one thing that I think is distinguishing our program. And John, you touched upon it, although it's come out in some prior discussions that we've had. Something that we pay very close attention to, especially with the human subject work, is the coefficient of variance of the assays. And you mentioned small sizes, small sample sizes, but it's also critical to think about how to avoid having to repeat the study that you've just successfully performed. And the strategy there is to make sure that your assays are validated prior to the initiation of your study, ensuring that you can really trust the data that's coming off of your technological platforms. And I know many of us have probably done ELISpot. I spent 10 years trying to validate ELISpots, and it's not possible across different laboratories to really see coherence with those assays. We've learned the hard way that, you know, the first thing immunologists do is put their blood on top of a Ficol column and separate the peripheral blood mononuclear cells. This introduces 35% coefficient of variance to your assays. So we've been working with a company to develop whole blood syringe-based medical devices that allow us to do functional immune monitoring without PBMC purification. We've eliminated the variability due to transport for the laboratory, and we have intra-donor coefficient of variance of less than 10%. Similarly, on the assay side, we're trying to focus on technological platforms that are going to give us reproducible results that are going to allow us to move forward clinically with any biomarker hit or mechanistic insight that we have in the context of the pathogenesis-based studies. And we've been utilizing quantitative PCR, which we're all using sort of in the lab, but with the Biomark open array system allows us to do sort of medium throughput analysis and protein assays on the Luminex platform, again, both with less than 5% coefficient of variance. And I do think it's important, especially in thinking about questions of translation, that we pay very close attention to making sure that the assays are in place prior to introducing them into the clinical trials, lest we have to repeat the same clinical trial once we think we have our first hit.
That I would like to raise, and it is really linked to the relevant parameter to follow. It is very important, and in the examples you have written on your slide goes very long with that, to look at the immune signature where we can look at it in the blood. But if we want to use an immunomodulator, which will be useful in cancer, for instance, will be useful in an inflamed joint, will be useful for multiple sclerosis, what is important is how does this immunomodulator affect the immune signatures, the immune compartment at the place where it is important, and which is very diluted in the bloodstream. So are you developing or people...
And I'm not saying that there's not a role for discovery-based science. Certainly the chip-based technologies and RNA-seq and all of the high-throughput omics technologies that are coming out are very helpful for coming up with candidates. But when you go into an interventional situation, you should already have that information about the disease pathogenesis and not try to both simultaneously develop a new therapy and a new assay.
Depends.
Is that, yeah, I was going to question this question. I could advocate a different approach, which would be indeed, if we really want to pass in immunology, what we are discussing today, from a general to a targeted therapy, it might be indispensable that we build together with the novel approach, the novel technologies, the novel target to assess it and to follow it. Philippe, I see you have the...
Yeah, I wanted to react to the point that the definition of the immune signature is rather obscure to me. and it can vary in time. And so there are, I think, different possible philosophies here. One is, and the one which is followed in SIGN, and I think John will comment on this after that. I think the approach we've taken globally is what I call systematic immunology as a basis for future systems immunology, which is not quite there. But basically, the philosophy is that when needed, you not only measure TNF-alpha and interleukin 17, you measure them all, point. And this is stored in databases which are built in such a way that the information is stored and usable for years and years and years.
My question is, so, where do you measure it?
Well, okay, no, it's part, but the comment is that if you measure as many parameters as you can with a given sample and with the available tools, you build a huge database out of which signatures are going to emerge or not emerge with time and may vary with time. But at least you can go back to your database and find things afterwards that you didn't think when you did the experiment.
Yeah, sure.
Okay. So that is very important, and then it becomes extremely technical, and maybe then John can talk about it, because then you need an accumulation of technology which has basically no limit in a way. So you have to choose the most important, but clearly it can end up measuring hundreds of parameters per sample, even if when you think of the clinical trial, you need only 10. Okay, so this is a different philosophy. I wanted to just emphasize this.
And Philippe, you're not wrong, and there are a lot of people that are taking this approach. And the idea of monitoring what you can is fabulous, and offering data warehouses that can be mined is an incredible opportunity. But pardon my French, but if you put crap in, you're going to get crap out. And yes, the hybridization-based array systems, your proteomic-based mass spec is going to be very good at finding things that are 100-fold changes, but they're not so great in terms of precision at finding the things that are 2 to 5-fold different. And we do have to be careful, because coefficient of variance on these assays is not something that is additive, but multiplicative. So in terms of integrating these large data sets, you have to be very aware as to how accurate your data is, so that when you do start integrating it, you're not being misled by some of the hits that arise.
I think that this can be addressed operationally again, and I think almost all of this stuff can be addressed by putting together an operational infrastructure that supports this. Separating technology development teams from run teams. The run team runs on validated SOPs all the way through for the given thing. Technology development happens with pilot studies, with smaller scale studies, where you actually develop the assay itself, and you ask, what can we ask about T cells? First of all, we already have a toolkit for human immunology. We've worked in the field for a long time. But can we develop new assays? Can we validate these new assays? Can we then put them up against gold standard assays that the run team is using to understand the dynamic range and sensitivity of that assay, and then move that over into the toolkit that we can use? Without a doubt, if you put in assays that aren't validated, if you take a given SOP and begin to manipulate it halfway through the trial, it's just nuts, and that's SOPs for processing, for any of it. And by separating these two events, that is technology development and the run itself and the analysis, this really does that. And they're on different samples too. I mean, you really do need a cohort that you develop assays on, essentially.
Just along this line, what is quite surprising sometimes, for instance, in the field of cellular immunotherapy, is that even the most straightforward monitoring techniques and approach are generally ignored when implementing a trial. For instance, one key issue in adoptive T cell transfer is the in vivo follow-up of T cells.
Sure.
And the problem is that currently we do not have techniques apart from the classical indium-oxynate labeling approach, which actually kills your cell or dramatically affect their homing properties, that would allow you actually to merely quantitate and follow up the survival of your T cells and their migration actually to the targeted tissue. So this shows that this is actually extremely important to take into account already before even designing the trial, the monitoring tools that are available actually to follow actually your product.
This I think is another example of this clash of cultures, that once you reach that stage of the trial, it's not about the science anymore. It's about the regulatory endpoints. That's all that matters.
Exactly.
Yeah, science is always secondary at this point. And this is where you need to come in during assay design development and build in that as a secondary research arm. Okay, so the primary endpoint is always going to be reg, but you need to fund the research arm independently of the trial itself so that you can assure that you're getting these samples in a timely fashion. You also have to run the entire team. So from the research standpoint, you need to run sample collection, you need to run processing, you need to run all of it. Otherwise, it'll just get lost in the mix.
Okay.
Next point.
Let's go on. So, next question, how can we provide proof of concept in order to secure massive resources required from translation? And how do we convince ourselves, and even more difficult, our colleagues, scientists and clinicians, that it is important to identify questions of common interest? So who is going to introduce this question?
Yeah, well, in this slide, I just wanted actually to emphasize some very straightforward points. Well, typically, the preclinical proof of concept of many immunotherapy trials rely primarily, if not exclusively, on in vitro data. And with respect to in vivo system, the mouse model naturally are extremely useful actually to get some proof of principle in various physiopathological conditions, but as you know, the transposability of mouse model to humans is quite poor. Not only because of species-specific difference in terms of innate and adaptive immune response, but also because merely the way a product is assessed in the mouse is very different from what actually occurs in patients. Just to illustrate that, for instance, in an oncological setting, well, you are using generally primary tumors. You do not even take into account, for instance, the previous line of treatment and the immunoediting of the tumor and so on. So that actually even if we would have actually a transposable immune system, actually the clinical setting is not the same than in the patients. So the point is here, how can we address actually these caveats? So naturally, well, one possibility is to use human xenografted mouse models. So in this situation, actually I would say the best model would be actually a mouse reconstituted with a human immune system, presumably, for instance, from precursor cells from patients, and human primary tumors with all the classical features of the in real life. But clearly, in this situation, we have still several limitations. Naturally, we can assess the pharmacokinetics of your product, but in terms of pharmacodynamics, then it's another issue, merely because, for instance, in a cellular immunotherapy protocol, the migration of your cell to the tissue might be affected. So this is a promising model, but the problem is that it's extremely tricky to establish, it's very expensive, the processing of this kind of model is quite limited, and I'm not sure actually that this can be the solution. So you can have actually other strategies, perhaps, for instance, to better adapt actually the therapeutical evaluation of your molecule that would more closely match what is actually occurring in a patient, for instance, by introducing lines of chemo or radiotherapeutic treatment in the mouse, although I'm not sure that it will work. So there might be also naturally the question of whether one could use a primate model or directly go to humans. So I think that typically here, this is the kind of very important questions that could be discussed now.
Yes, please.
You had a limitation, and the topography of your organs are really different.
Yeah.
And it may be very important. And just to add this, because we personally in our lab, we developed some transgenic pig. And for instance, we have developed the pig to study cystic fibrosis, or to also to work on the macular degenerations and all that stuff. But even in that case, you know, it's extremely difficult and it's extremely costly to work with such animals, transgenic animal, because of the development of such animals, and also how the people can feel this. And we have the José Bové of the pig around us, and we have, of course, a flying gene. And to develop such pig, we need the A3 facilities, and to develop a colony, it's really impossible.
Well, there is naturally, the question, the recurrent question of the bench to bedside to bench to bedside is depending actually on your clinical experience and better knowledge, actually, for instance, of the oncogenic pathways, primary or secondary oncogenic pathways, or your patient samples. Then you can try to reproduce these pathways and design a mouse model that would better mimic the human pathology, which is naturally an obvious solution, but it's a lengthy one.
Can we be a little bit even more provocative? We were discussing regulatory needs, and it is clear that there is a contradictory situation in Europe, at least, which is one, animal experiments should disappear and be replaced by in vitro analysis, because this is we shouldn't hurt animals, and we should try to find other ways to mimic or to model what we do in animals. And at the same time, there's an absolute requirement of animal models as a proof of concept for any new development of a drug. Couldn't one say that obviously an animal model can give you what is in the model? No more and not less. Indeed, if you analyze cytostatic agents in cancer, nude mice grafted with a tumor, okay, you will see if it interacts with DNA replication or not. If you want a PI3 kinase inhibitor, and if you have this driving modification, it's fine, you can use it also. Once you go in more sophisticated situations, such as immunomodulation, it is fine to do physiopathologic analysis, but as you said, they will improving and refining and improving again the model, trying to humanize it as much as possible, and you will not humanize the blood endothelial cells, for instance. You will have capillary leak syndromes that you have not expected, etc. Will always be, in my view, running after doing better, but with a big hole between the two situations. So I would advocate the position to say, let's ask the simplest questions as we can to the mouse model. Let's accept that there will be a gap, and try to do in vitro as we can, not to make a crazy thing when you pass in human. But there will be no way. I don't see how we can do that better and better and better, and always we will be in a poor situation.
Pragmatically, it's exactly what occurs, actually, what happens. Is that basically, the regulatory agencies, they consider that, once you have made significant effort in getting the strongest preclinical proof of concept well, they say, okay, so then in this case, just design your trial so that actually the risk-benefit ratio will stay positive.
There's one example which is very well known in immunology, I would say. I think the number one drug, let's call it a drug, sold in immunology should still be rituximab, more or less. Rituximab, by definition, couldn't be tested in mouse model. It's certainly not for collagen arthritis or for anti-DNA antibodies or in all the autoimmune settings that is now the developmental field for anti-CD20 antibodies. So I don't know, maybe there are reactions, mouse people in the audience, maybe there are reactions. I do not have the feeling that we should, I think, although I fully agree with the fact that we should have better infrastructure, better immunomonitoring, trying to do it on smaller samples, costly effective, answer the question, how much can we refine mouse models to ask physiological questions? Clearly, this is an important matter. But to ask translational questions, more that I would simplify the model. More that a simple model can answer, I have the feeling that it is a lost run. But maybe there are other, everybody agrees with that, or everybody is just fed up with Sebastian,
so
maybe
I can
an idea on that?
Maybe I can
You?
Maybe I can continue. I mean, the mouse is going to remain a very important tool to ask scientific, mechanistic questions. But if you're interested in human, that's sort of what we were challenged with. I just want to offer one sort of counterpoint to what Marc proposed. And I don't think I knew it with forethought, but maybe it was just being trained as a clinician or the specific medical questions that we were interested in, but we've actually been doing it from reverse, and bench to bedside sort of cropped up as a catchphrase about 5 years ago, and I was sort of playful in the idea that what we were doing was really bed to bench, and we've been thinking about this as now sort of reverse translation, and I just wanted to offer one example of what we've been doing in the lab over the past few years. And this is in the context of hepatitis C, where arguably we don't really have a possibility for a mouse model, spare some of the very sophisticated hep-Q mouse models. Chimpanzee are the only animal model, in fact. So we've been working from human. We're interested in human disease, so we start with humans. And what's very unique about hepatitis C is that we have a positive control. We treat patients with interferon-based therapy, and half of them fail to respond to therapy, half of them do respond to therapy, and that's been very important from the perspective of being able to do science on human populations. And in the context of some of our biomarker discovery, we identified IP10 as a negative predictor for response to therapy. And what took about 5 years to really deconvolute, starting with the clinical observations, going into very strong biochemistry and mechanistic studies, we understood that what was happening was that IP10 was being produced by the liver, but that there was also the upregulation of an enzyme, dipeptidylpeptidase 4, that was cleaving the molecule and converting it into an antagonist. And we've now moved this into mice with a very specific mechanistic question of understanding, does DPP4 regulate lymphocyte trafficking? And we could look in the knockouts, and we could do all sorts of things to mice that we can't do to humans. And what's really quite gratifying is that we're now in a position with small molecule inhibitors to ask what's going to happen if we block this enzyme, restore the chemokine gradient, and can we use this as a strategy for enhancing the interferon-based therapy efficacy in the context of chronic hepatitis C. And I don't know if it's long or short. Certainly for the people involved in the work, it feels very long. But if you think about the initial biomarker hit being only 5 years ago, and the fact that we are just now opening our phase 1/2 interventional clinical trial with a small molecule that's actually quite short. And I do think that as far as the question of translation, starting from disease pathogenesis and working backwards may be very helpful, certainly satisfying as scientists because we're understanding mechanism, and can lead to some relatively surprising insights into how to go back to the bedside with new theranostics or new therapeutics.
But typically in this situation, actually, the probability here of success is greatly increased because clearly you start from a better knowledge of what is going on in the patient and you translate it to the murine models. So.
And you ensure that the mouse is mimicking the human,
Yep.
rather than trying to take a mouse observation and try and
exactly.
force it into a
I
human
agree.
situation.
Okay, should we address the next question, which is essential? Current model of drug discovery, whatever the drug is, and utilizing of the drugs are of course made by how this interact, how can we play with this, and I think that you're an expert.
Okay.
in this kind of thing.
Thank you. Great. So the Singapore Immunology Network is part of the Biomedical Research Council at ASTAR, and ASTAR falls under the Ministry of Trade and Industry in Singapore. And so really I think this is just a recognition that if there's a medical need, then there's an industrial interest. And so obviously some of the major drivers of meeting medical needs, is, as we've just heard, is a fundamental understanding of the basis of disease. And so we've got a number of models that go on at SIGN for the immunomonitoring platform and for the overall program, which includes really co-discovery, co-development, R&D interactions, particularly the ones that are really favored are really co-development interactions. So working with industry to try to find the fundamental underlying driving factors for a given disease. And we use and apply the technology that we've spoken about to look at clinical trials. And so because we're under the Ministry of Trade and Industry, there's obviously a mandate for things like out-licensing, technology development, and a number of other things. So the reason that I think that this is important is not, is really because when you think about the number of people that you actually have, there's two aspects here. One is the size of the clinical trial that's required to actually address the questions that you're asking. So whether it be the molecular nature of vaccine protection in humans, or understanding fundamental questions in cancer immunology, you really require a lot of patients. You require the infrastructure that we talked about, and a lot of this requires an enormous amount of team-based research. So you need the funding to do it. You need the institutional support to actually do this. And so partnership is a necessary part of a lot of this. And so to do that, you really have to look at what you're doing now and ask how can we create something of mutual interest with industry. And there are a number of models that can move forward. And one of the key parts of this is understanding what your partner wants. And so oftentimes you're both scientists and you both are very interested in the scientific questions, but institutionally, the institutions themselves have completely different mandates. And so understanding upfront what the key questions are and then putting together an agreement that is of mutual interest to both parties, so that you're not essentially using, say, an immunomonitoring platform to monitor a clinical trial to simply address biomarker discovery for an interesting biomarker. You absolutely have to have a research interest that addresses an interesting scientific question, as well as something that is key to the industry, which of course would be the development of a new product.
John,
Sure.
I feel terrible having this discussion at Collège de France.
Yeah, I do too.
And I just wanted to ask you about your particular institution. Science started out as a pursuit of a philosophical thought, and then sort of with Claude Bernard and others, a question of humanitarian interest. You and I were probably both trained in that sort of idea that science was about for the benefit of humanity. You're now at an institution where it's for the benefit of economy.
I disagree. So I think that as I said before, that first of all, science is something you do, not something you are. So you're doing science when you're asking fundamentally important questions. And so what we do is we look at the landscape and we say, and so it's the same with the medicine and the clinical trial. So we look at the landscape and say, what do we need to actually ask this question? So what we need is 10,000 people, and we need longitudinal follow-up, and we need all this kind of thing. So who is doing that right now? Who is collecting 10,000 people? Well, there's a vaccine clinical trial here. This is an interventional study. This is a study in which I can ask a scientific question, a before and after immune perturbation event to study human immunology. Okay, so that's the scientific question that we're applying to it. Now, you have to understand and respect the primary driver of that trial is the development of a product. And that's okay, and that's all right. But that doesn't mean we can't still ask fundamental scientific questions on top of that by applying, what we know to the actual trial. So I don't think that there's I absolutely don't think that there's, we really go head to head with each other in terms of our goals. And in fact, of course, we synergize in the sense that, and I'm not saying this, you hear these words all the time in industry, like synergize, but I'm saying this in the real sense that we identify things that industry really can't do. So we do things like, creative destructive process where we try things and then look for, and what we find is by looking for the fundamental mechanism underlying the concept of protection, we take that information and come back and rationally redesign the vaccine. So I think that it's actually a very good partnership between both industry, medicine, and science.
Have you felt any limitations at SIGN because of your being under the Ministry of Economy?
Mostly expectations, actually, to tell you the truth. So I don't think there's any. No, I think it's enormous opportunities because of this idea that there may be something that comes out of the back end of this, that you get an enormous amount of support from everybody. But the expectations and the timelines are something that I'm not sure everybody understands. So that the timelines of understanding a fundamental discovery to all the way to to a product. This is, in biomedicine, this is an enormously long thing. And actually, I got to say, I think this is a failure of academics as opposed to a failure of industry. So that, the academic model of taking a postdoc and giving them 3 years and then saying, okay, good luck, you're assistant professor now, there's nothing you can do in a clinical trial in 3 years to realistically get anything out. They have to walk in right toward the end and do something innovative. And so I think that this human immunology in and of itself really kind of has to change away from sort of an academic training model into something that can accept more of a broader and more team-based approach.
Philippe.
Yeah, I think I can answer your question in 1 or 2 sentences, namely that SIGN was designed as a small Pasteur Institute. A small Pasteur Institute. So a Pasteur Institute is dependent on revenues from industry as well as from the charity of the public. So far in Singapore, there is no charity of the public, but clearly we target about the same percent of revenues from industry in time that we have in the Pasteur. Pasteur being the benchmark for excellent basic research, compatible with a productive interface with industry. So this is exactly how it was done. Now, let me go back to the issue of industry with 2 points. Point number 1 is that there is an enormous amount of expertise in industry, which you don't access if you don't talk to them, okay? So as having good relationship is very important because you learn a lot, especially when you go, of course, to the interface between the clinics and treatments. So this is important in that respect. The second point of clarification I would like to make is that clearly for institutions, academic institutions at large, including Pasteur, SIGN, Nantes, and others, all academic institutions, or most academic institutions, the problem is to know where to stop and not go too far away in in having an industrial partnership. So to give you one example, and that goes back to what Jean-Pierre said some moment ago. Clearly, when you develop a product, okay, you develop a product which is not exactly the same as immunotherapy, because it's not a product like the other product. It's not a drug and it's not a vaccine. Well, clearly, industry is going to take care and has to take care of a large-scale clinical trials. That's for sure, okay. The problem, though, is that industry in the last few decades has done relatively less research clinical trials than before. And then the gap has been created between academia, which didn't have the resources to run the clinical trials or research clinical trials, and industry, which was targeting blockbusters, okay, and was willing to do phase 1 clinical trials only if there was a blockbuster in the end as a significant probability. And I think this has killed part of the innovation in industry. This is my straight assessment. And I think this has been an enormous mistake. Now, a number of academic institutions, starting with NIH about 10 years ago, 15 years ago, have started equipping themselves to be able to substitute for the research part. And academic institutions at large are now trying to do that. My final point will be that when we talk about clinical immunomonitoring, it's not only the clinical trial. It's the analysis of samples, whatever you get. And of course, you can get a lot of information from biopsies, for example, and therefore accumulating information about these samples is also part of the immunomonitoring. And this is research. This is really research to help the clinical research.
I think this is a major question, and it's worth discussing. However, it's clear in my view that we are discussing translational research. We are not discussing science as a whole. So if we want to translate, we need to translate to somebody, and we need to have the people who will be the real translators to the last step as early as possible in the field. And somebody was, I don't know who of you was discussing the fact that once it is in the clinical trial, and in other words, in industrial ends, the questions of the immune modulation, for instance, disappear. And I think this is a big problem. I remember having been a counselor in a company that Philippe knows very well, was trying to develop vaccines to treat cancer. And I was saying, you know, what should be done is really to do trials in which you compare various doses, various dosing schedules until the point that you show that you have induced a vaccine effect, that you have an immune response to the vaccine. And then you go to the clinical trial on prostate cancer and whatever. And the answer was very clear of the other members of the board as well, saying, no. If we have a positive effect, we don't need that. And then we'll see afterwards. We will decorate it with your stuff. And I think that if we are not present as basic scientists, very early, you even wrote co-discovery. Yeah. That is sometimes difficult, but still, if it's possible at this level. But at least very early in the process, to influence. We are so afraid that they will influence us, but let's try to influence them with the fact that there are questions that must be approached in a science-based way if one wants to go a step further with efficacy. And I think this is our role.
But the paradigm has changed a bit during these recent years, and clearly now even the big pharma realize that they really need permanent interaction with academics, simply because actually now the business model is not based on huge blockbusters that would target actually a very large patient population. But now that the pharmaceutical industry has realized that actually you have to go towards personalized medicine, then you really need actually to better understand the underlying mechanism. So this needs basic science in order actually to identify, for instance, the markers that are predictive of therapeutic efficacy. So, yeah, exactly. So you need actually an excellent clinical annotation, you need actually an excellent biological monitoring, and you need actually an excellent understanding of what's going on. So. This slide is clearly in line with what Philippe just mentioned. Actually, the point is that when you are developing actually a clinical trial, it's a lesson of pragmatism and humility. And really here, the interaction with the industry is extremely useful. They are very pragmatic. And as highlighted by Philippe, actually, we can learn a lot from the industry. Because they have the way, actually, of readdressing what you have done at the academic level in a presumably much more rigorous fashion. So, in particular, actually, what they are supposed actually to perform all this with good traceability and the GLP condition and so on. So what is very important is that, and it's typically the problem of the gap between actually the clinical trial and the preclinical proof of concept. In the negotiation with the industry, you need to get actually very strong argument in favor of not only the efficacy of your molecule, but also limited adverse effect, good pharmacokinetics, and so on. And the problem is that to get all these parameters, you need actually, it's very expensive, you need actually a huge number of facilities, for instance, just for instance for antibody-based therapies, what we crucially need is generally good histopathologists that will perform actually all the screening and guarantee actually that your antibody is truly specific to the targeted pathway or molecules. So even this actually is key actually during the negotiation with the industry. But also I think that what is useful when you get, when you interact quite early with the industry is that they have generally the mean actually to screen a large variety of systems in order to identify possibly clinical indication that you didn't thought of when you performed actually your experiment at the academic level. So I think that typically here, I have just mentioned here the various points that are key actually to strengthen the proof of efficacy and the chance of success of the molecule, because remember that the attrition rate is more than 99%. So the objective here is to kill as early as possible candidate markers without before investing too much. So clearly here, what is important is GLP infrastructures, is broad access to physiopathological models, get very rapidly data for toxicity, pharmacokinetics, possible side effects, and so on. And so one question is that since it's very expensive, you have actually two possible models that are actually currently tested in France and in other countries. Is either you establish large networks that would involve academic and industrial partners, or you massively fund actually some purely academic networks that would have actually the advantage of assessing some combination that might not be, that could not be tested in the industry, merely because, for instance, of intellectual property constraints. So. No, we have plenty of things to discuss.
Last slide until we come to philosophy. Because the last question that we have in debate for the last few minutes that we have left is something which I think is relevant to the Collège de France, and is what is normal and what is not normal. And It could have been also normality is one question. Else could be another question. What is else as opposed to disease? Is normal what is not abnormal? Is else which is not sick? And how can we translate our finding to disease populations without knowing what is normal? This is a question they have decided to ask. I could see very easily how we can do that without knowing what is normal, but I think it is a major question. If somebody can answer that question, I think that we will be a benefactor of humanity. But
so it's John.
Sure. So I'll open up.
You know.
Yeah, I do. So I'll open up by simply saying if one wanted to ask what is normal, probably the worst system to look at is humans, because they're extremely difficult, as we all know. One of the reasons that we don't look in people is that, and we stick to inbred mice, is that there's an outbred population. They're difficult to deal with. They object when you take their spleens. They're expensive, and really the need for understanding what, particularly on clinical trial, in human immunology studies, what the normal levels of, say, serum TNF receptor are in the population is extremely important, because when you're analyzing either clinical samples or you're getting onto a clinical trial, there are ethical constraints and obviously the kind of controls that you can actually run in the experiments you have. So there's an enormous need for a deep immunophenotyping database that can be constructed with well-curated normal donor control populations. And so applying the technology that we talked about to normal donors, along with some of the genomic technology that's out there right now, can give us a baseline phenotype in which we can then compare these samples to. And as of right now, this doesn't exist at all. And so what we really need to do it requires a lot of collaboration, actually. So to create our own cohorts, but importantly to share SOPs between the cohort groups, including internal quality assurance standards. And so this really falls under this concept of biobanking. And to utilize the immunomonitoring technology that we've developed to monitor longitudinally clinical trials to phenotype these individuals. And so I can say that at SIGN we've got an excellent cohort that I think it's really not reflected elsewhere in the world, in the sense that it's a model by which the immunomonitoring platform has collected a wide variety of samples, but investigators can then access those samples and run particular assays, as long as those assays are done on the entire cohort, and that data can then be shared. So you'd have an expert in T-cell analysis that could then work with the immunomonitoring team to put together, say, a T-cell assay for their particular disease of interest. That data can then be dumped back into the database, so we can, over a period of time, have a deeply immunophenotyped cohort of individuals that we truly understand both the serum proteomics, the response to TLR agonists, the pathogen history, everything about those people, and can then mine that data for quite a while. And not just for an understanding of those individuals, but also to establish a baseline for comparison with sick individuals.
So it's been another area of very exciting interest between John and myself. And
So just a question on that too, Matthew. So do you guys have any banking capability? That is, one of the things that you run into when you look at the older banks is that whether or not you can anticipate future technology development, and you wish you had that panel, a couple of years ago. But so is there a banking on this?
There is some banking. We will have aliquots from the supernatant and from the cells that stored.
Okay.
We are thinking obviously about epigenetic approaches as those technologies come online.
Right.
Metabolomics as those assays become more refined. But certainly we're a little bit limited in terms of the amount of blood that we can take from these healthy donors. What is going to be an incredible opportunity is the renewable IPS lines.
Yeah.
And as our sophistication grows with respect to how to generate different generations of IPS, we'll be able to go back to fibroblast lines. And then obviously the capacity to differentiate them into cells of interest will enable tertiary screens.
Right.
And they're genetically annotated, so it's interesting to be able to sort of immediately go into the genotype to phenotype analyses.
The number of 1,000 LC donor has been chosen because it is what you have the money for, or in view of the immune system diversity and polymorphism as a other basis.
There's no math yet to do power calculations.
Yeah, right.
The types of studies that we're thinking about. The largest QTL study that has been published thus far is 85, which is the argument that Luis offers when challenged in that way. We're doing this actually stratified, half men, half women, 5 decades of life.
That is important.
5 decades of life, from 20 to 70, so that we'll get some sense of what's happening for aging. So there are a number of different ways that we're thinking about parsing the data set. We don't know. We think that we'll be able to have depth in our cohort to find things that are 5% variants within the SNP genotyping variants within the human population that are responsible for 50% of the immune phenotype. But understanding the math behind the different interactions across different SNPs is still something we have to discover over the next 10 years.
I think there's a fundamental, and I know we've talked about this before, but I think there's a fundamental difference between the two approaches that we're taking. One is that this looks a lot like the UK Biobank in a way where you're really, you're set on the methodology that you're going to use, and you go for it across a large population in order to address a particular medical question, for instance, and powered in a particular way. But I think the approach that we took is a lot more academic in nature, in the sense that what we said was, let's collect X number, 1,000 people, bank them, and then open this up for the purpose of human immunology research to experts in every field of that research, whether it's a vaccine question or whether it's an allergy question or whatever. As long as that the samples are run in a professional manner under SOP, and that we get very good data quality out, and that the banking, the draw and banking itself is done in a professional fashion, then once we pull this data together, we can make new discoveries. And that this is essentially, from a scientific standpoint, paper-driven and discovery-driven. And that's really how it comes down.
I mean, I think that's correct. There's one significant difference between what we're doing and what the UK Biobank is doing, which is the idea of stimulating the immune system.
Okay.
And this allows us to use the individual as their own point of reference. And I think that that's going to offer a lot more power to the types of data that we're getting out. And in fact, that's where our experimental ideas are integrated. And I don't obviously have time to go through the entire concept behind the program, but one of the things that Luis has really been remarkable at in terms of innovation is understanding how, through comparative genomics, understand which single nucleotide polymorphisms are under positive or negative selection. And it was quite remarkable in applying this to some immune genes, where he did gene resequencing of all TLRs and understood that 3, 7, 8, and 9 are under constraint, and human population does not allow variants in those genes. 1, 2, 4, 6, and 10 are under genetic drift. suggesting redundancy. And 5 is under negative selection. 25% of human population, or I should say Europeans, have dominant negative TLR5. So that argues that probably flagellin is not the best candidate for adjuvant vaccine development.
Sure.
When I saw this data, I was aware that we had to do this for every pathway, so that we're studying the genes that, and the single nucleotide polymorphisms that evolution is indicating are relevant, and not the others, in that we do have very short careers as academic scientists.
Yeah, it's a great case for sharing, because I think we're running similar experiments, of course, with panels of all toll-like receptor, all nod-like receptor, and looking at the output. One question I do have, though, is, so I think people can put these kind of studies together, particularly if they're in an academic medical setting, or if they're, more importantly, they're in a university where they can round up a bunch of students. But how do you address some of the, I hate to get to it, but ethical and regulatory problems that come with running a study that has broad and future consent, where I'm going to draw samples from you, I don't know what I'm going to do with it, and it depends on technology that hasn't been invented yet?
I'm getting signals from Hervé that we're bumping into 6 o'clock, but just
we are approaching
as an American working as an American working in France, I can say that I'm really delighted to be running this project in France. Some of the advisory board members that we have couldn't believe what we are able to do in France, and I think that that has to do in particular because of the medical legal perspective of human subject studies. We have a lot more leeway here than exists in the United States. Okay.
So we arrive at the end. Is there let's go directly to the take-home messages.
Well, just to end this session and just to insist on the fact that really, and we have repeated that throughout this session, is that we really need, when implementing this kind of clinical trial, pragmatism and a lot of perseverance and patience. And clearly, while it's a lengthy process, the complexity and cost are generally underscored. And I would like just to mention, it's not very practical, some keys for success is clearly, well, we have to prioritize simple and robust protocols. This kind of trial require actually really a transdisciplinary team approach that should involve clinicians, experts actually in cell developing, in cell therapy, in regulatory filing, private companies that can be important money providers. One point that we didn't have time to discuss is the question of disease indication and what are the relevant criteria for disease selection. And I should insist on the fact that one key point when selecting a disease indication is clearly to realize that we really need early surrogate endpoints in order to have a rather short time for assessment of clinical response. And also, we have to be aware actually of the recruitment constraints, particularly because this can impact actually on your duration and cost of your trials. So you have to be aware of the incidence of the disease and more importantly, the existence of competing protocol. And this is generally a key issue, for instance, in oncology, because it dramatically hampers your patient recruitment. Also, one last point, and we mentioned this, is the question of monitoring. So you have to have in mind at least how you will monitor your patients, either through candidate approach or through global profiling, with all the caveats actually of global profiling, and you really need to get or to develop specific monitoring tools that will allow you to get as soon as possible predictive markers of the protocol on therapeutic efficacy. Thank you very much for your attention.
This session in which we have tried to address several of the questions which are important for translational research, try to make it a little bit better. I'm sure that we haven't answered all the questions. I have noticed one thing, is that we started with translational research and we got all excited when we came to basic science, because the question of normal immune system is really basic science. And here people began to discuss TLR5 polymorphism or thing, and the excitement, and if I wouldn't have been very strong, they would still be discussing about these points. And when I was listening to those, I had the impression that the discussions that we have had during this hour and a half is really a tribute to Philippe Kourilsky. It's difficult. I think I'm the only one of the same I was going to say generation, more or less, in this room, and I have known he is the perfect summary, I would say, of what we have discussed. I have known him, he was a phage specialist at that time, a phage expert. And then he went from the phage expertise to chicken, but not so much to do. I don't think, Philippe, that you were thinking of having a nice antigen for it, for studies in oncology, but you wanted to study gene regulation and what was important in that. And then he jumped to difficult MHC, gene diversity, polymorphism, and how all of this went on the molecular level. And the same gradually, Philippe also did take the Rennes, or do you say, of a major institute in France. He was a half-time director in industry. He has created, with Paula and a few others, another institute. But as we did at the end, the impression I have is that what makes he and him and us, I think, very open is when we come back to science and when we have open eyes. And in the take-home messages, together with telling by friendship, and also my thanks to Philippe, who has been a very quiet and secure figure for many of the people, not many still remaining here, but during all these years, including myself for a short period. I would like to add one take-home message. If you want to have the things going more rapidly and better, do as Philippe does. Keep your eyes and your ears open, and when you see something which looks exciting, go after it, and then it will enter in all of these infrastructures, superstructures, normality, disease. I'm sure there will be a lot of very prominent people that will be able to make these things work. But having the open eyes, this is, even for translational research, is something which remains essential. With that, I will leave you and thank all of you to have stayed with it. It's raining, I'm sure, outside, so you can stay 1 minute later more to have spent this time with us. Thank you very much.